In this unknown lab, everything looks controlled, measured, contained, understood. But what is being studied here is not ordinary life. It is built from the mirror image of the molecular building blocks used by every organism we know. Its chemistry would be reversed, its proteins reversed. The machinery of life itself rebuilt in reflection. Our immune system would not recognize it. Humans would not recognize it. Nature would be terrified of it. But what if this stops being hypothetical? What if we lose control? Many molecules come in two versions that are mirror images of each other, just like your left and right hands. In chemistry, this property is called chirality, from the Greek word for hand. The amino acids that make up proteins, the sugars in DNA and RNA, and other biomolecules all come in left-handed and right-handed forms. Homochirality means that life has picked one orientation and stuck with it. The DNA of all known organisms uses right-handed sugars, while proteins are built exclusively from left-handed amino acids. Why nature chose this configuration is still a mystery. A NASA-funded study published in late 2024 showed that early RNA enzymes can work with either-handedness, suggesting that the preference may have emerged later through evolutionary pressures. The asymmetry is so ingrained that a right-handed amino acid simply doesn't fit into the machinery designed to process left-handed ones. It's like trying to shake someone's left hand with your right hand. That incompatibility is exactly what makes mirror chemistry both exciting and dangerous. Over the last few decades, chemists have successfully synthesized mirror versions of individual biomolecules. These include mirror peptides, short chains of right-handed amino acids that resist degradation and evade immune detection. Because digestive enzymes do not recognize them, mirror peptides stay active longer and can be dosed less frequently. They also cause fewer allergic reactions because the immune system relies on very specific chiro interactions. Pharmaceutical companies already use such mirror molecules in experimental drugs, and researchers see them as a way to create therapeutics that last longer and are more stable. Some synthetic biologists have imagined going further, building an entire organism from mirrored components. A mirror bacterium could churn out mirror molecules for us, making large-scale production of such therapeutics more efficient. Other proposed applications include mirror-based materials that resist breakdown and infections, or even exploring whether mirror life could exist elsewhere in the universe. This ambition to push the limits of synthetic biology is part of what makes the mirror life story so compelling. Creating a living cell from scratch is extraordinarily hard. Yet scientists have made impressive progress toward mirror life. In recent years, they have chemically synthesized mirror versions of key enzymes, such as DNA polymerase and RNA polymerase, and built pieces of the protein synthesis machinery. In 2024, researchers published a technical report detailing the feasibility of creating mirror bacteria, and estimated that if an international consortium made it a priority, a self-replicating mirror bacterium might be achievable within 1 to 3 decades. The drive to build a mirror cell isn't just scientific hubris. Developing the ability to stitch together entire genomes and ribosomes, mirror or not, could accelerate our understanding of life and unlock powerful tools for medicine and biotechnology. But that dream comes with a shadow. In December 2024, a group of 38 scientists, including Nobel laureates and pioneers of synthetic biology, published a commentary. They warned that creating a self-replicating mirror organism could pose unprecedented risks to human health and the environment. Here's why. Immune invisibility and unchecked infection. Immune systems recognize pathogens by their molecular shapes. Because mirror bacteria would use reversed chirality, our immune receptors would likely fail to detect them. Commentary notes that mirror organisms could cause fatal infections that are largely refractory to immune responses. An evolutionary biologist told The Guardian that such organisms could evade many human, animal, and plant immune system responses and cause lethal infections that would spread without check. Ecological disruption. Natural bacteria are kept in check by predators like bacteriophages, viruses that infect bacteria, and protists. Those predators are also chiral. Mirror bacteria could escape them. Analyses warn that mirror microbes might replicate unchecked, avoid natural predators and antibiotics, and potentially evolve to consume ordinary food. Persistent populations could spread between ecosystems via animals or humans, threatening plants and animals and destabilizing food chains. Lack of effective countermeasures. Existing antibiotics, phages, and immune defenses all depend on chirality. A mirror bacterium would not be susceptible to them. Even engineered biocontainment measures might fail if the mirror organism evolves or escapes into the wild. Irreversibility. Once released, mirror life could be impossible to recall. As one expert put it, once a mirror cell is made, it's going to be incredibly difficult to try to put that genie back in the bottle. Despite the risks, mirror life research attracted talented scientists because of its scientific allure. Some authors of the 2024 report once dreamed of building mirror bacteria, but changed their position after analyzing the hazards. Their commentary emphasizes that mirror therapeutics and other non-replicating mirror molecules are valuable and should continue, but draws a bright red line at creating self-replicating mirror organisms. The report's lead authors argue that the world has a narrow window to set global governance before mirror life becomes feasible. They call for an international forum to define clear red lines, strengthen safety practices, and ensure that funders will not support the creation of mirror bacteria. Policy experts note that this is a rare chance to prevent a risk before it emerges. Unlike nuclear weapons, mirror bacteria do not yet exist, and the scientific community still has time to decide not to build them. Some scholars caution against letting fear stifle innovation. Mirror molecules already yield life-saving medicines and stable biomaterials. Careful research into mirror enzymes and non-replicating systems can advance fundamental science and lead to safer therapies. The key distinction is between using mirrored chemistry to build molecules and building self-replicating mirror life. Mirror life sits at the edge of science fiction and reality. An unseen microbe that slips past every defense isn't just unsettling, it's something evolution never prepared us for. And yet, we're getting closer, not out of recklessness, but curiosity, the same force that drives all discovery. But this time, the stakes are different. Because once something can replicate, once it can evolve, it may no longer be ours to control. So the question isn't just what we can create, it's what we're willing to risk. Because this might be a mistake we only get to make once.